All right, believe it or not, here we are in our last unit, Unit 12 on human remains. So hopefully you've been enjoying the course so far, and things will end on a very high note as we address this very final unit. Okay, so the first professional we'll discuss with respect to human remains is the forensic pathologist. This is a physician who has specialized training in determining the cause, mechanism, and manner of death. So this person would have gone through 4 years of undergraduate study, another 4 years of medical school, and then at least 3 to 4 years of a pathology residency, usually focused on forensic pathology at some point in that additional training beyond med school. And so after all of this, the forensic pathologist is trained and ready to conduct forensic autopsies, which we define an autopsy as the medical dissection or examination of a body. And if you recall when we talked earlier in the semester about medical examiners are typically forensic pathologists by training. Okay, a forensic pathologist doesn't have to be a medical examiner, but a medical examiner is usually a forensic pathologist. And sometimes in areas like large cities, you'll have one or more medical examiners, and then you'll also have forensic pathologists who assist with forensic examinations of bodies. Now, if we want to get at the beginnings of forensic pathology, we have to talk about the beginnings of modern pathology itself. So in the late 1800s, Rudolf Virchow discovered that diseases could be diagnosed by examining cells. So he gave credibility to the so-called germ theory. Again, before that, it was mostly sort of just an amalgam of religious beliefs that people got sick because they angered divinities or so on and so forth. But Virchow is the one who showed that it's actually possible to diagnose a disease by examining the cells themselves, and for this he's become known as the father of modern pathology. So again, if we think about the responsibilities of the forensic pathologist, they determine the cause, manner, and mechanism of death. So when we talk about the cause of death, we're typically talking about either a disease or an injury that resulted in the death of that individual. The manner of death talks about the reason for that death. So was it an accidental death? Was it a homicide? Was it a suicide? Was it just natural causes, right? Disease that caused death is a natural cause, so on and so forth. If we look at the mechanism of death, this is the immediate physiological breakdown of the human body. So, if we're talking about a homicide, was the person stabbed and they lost so much blood that they died? That would be the mechanism of death, exsanguination we call it, or by a gunshot. Again, they could have bled to death, exsanguinated from that. So if we follow that example of a gunshot wound, the cause of death would be the gunshot. The accidental death could be accidental again or homicidal, depending on the nature of that gunshot. Was it gunshot because the gun went off accidentally, or was it intentional and either suicidal, the person intentionally shot him or herself with the gun, or homicidal, they were shot by another person. Finally, the mechanism of death, in this case, we'll say it's cardiogenic shock. If the person is shot through the heart, they could have been shot through another part of the body and just bled to death, again, as I mentioned earlier, and then that would have been exsanguination as the mechanism of death. So all of this is dependent on the particulars of the case under study, so we just have to realize in general what the terms mean and then apply them specifically to the case under investigation. Once a person dies, their body goes through 5 stages of decomposition. So we'll take a moment to look at each of those stages here. The first stage that we'll discuss now is the fresh stage, and it's fresh because this is the stage that occurs immediately after death. So some things that happen upon death, well, the blood begins to pool to the lower parts of the body due to gravity, right? And we'll talk about this as livor mortis. The reason for this is that the heart's no longer pumping. There's nothing acting against gravity, right? So these areas where the blood settles become purplish or reddish or bluish. And this happens pretty quickly. It begins as little as 15 to 30 minutes after death. It becomes permanent after 8 hours. So if the person is in a state where they have these livor mortis spots, right, the purplish, reddish or bluish deposits of blood where there's gravity pulling the blood down and nothing interacting to keep it up, then we would expect to have that permanent after 8 hours. So we could use that to determine if the person's been dead 8 or more hours, or if they've been dead less than 8 hours, depending on the livor mortis appearance. We could also determine if the body's been moved after death. So if the livor mortis doesn't match up to the way the body's found, then that indicates that the body's probably been moved after the 8-hour mark, where the livor mortis became permanent. Finally, in that fresh stage, we've also got an aspect called pallor mortis, where the body becomes pale in color as livor mortis occurs, right? As the blood drains, according to gravity, it's going to leave the rest of the body a very pale color. And so we have in that fresh stage both livor mortis and pallor mortis as obvious stages of decomposition. Now, if you're squeamish like me, you might want to close your eyes or look away because we have a graphic picture coming up. If you don't mind that, you've got a strong stomach, then go ahead by all means. Keep looking, and for those who would like to look away, I'll let you know when it's time to look back. Okay, so here we have an example of a livor mortis pattern. And so in this instance, wherever we have those pinkish spots, those are where the blood had pooled, and where the skin is more flesh tone, again a little paler because of that pallor mortis, that's where something was pressing against the skin and not allowing the blood to pool in that area. So this particular pattern here is actually caused by the victim lying on an egg crate foam mattress, right? These were popular a while back, and so wherever the top of the foam mattress was pressing on the skin, you don't see the pinkish color. Wherever it was more of a divot there and the blood was allowed to pool, that's where we get that pinkish color. So that's what's responsible for the livor mortis pattern in this particular picture. Okay, we're no longer looking at that picture, but we do have an image of the bloating stage. Again, it's not particularly graphic, but if you don't want to look yet, you don't have to. In the bloating stage, gases build up and give this swollen appearance to the deceased individual, and that's resulting from the bacteria living inside the body that are producing these gases. The bacteria are still alive, and they've got plenty of food and fuel to undergo metabolism, so they produce these gases that aren't able to escape, and so first you'll get bloating, and then eventually you'll have the body leak these fluids from orifices because a sufficient amount of buildup has occurred. So that's our second stage of decomposition, and it's known as bloating. Okay, so no images on this slide. Figured I'd give everybody a break here. And our third stage of decomposition is known as active decay. So in active decay, bacteria and maggots actually break the skin, and so you get an unpleasant odor that escapes and is notable, right? Humans do not like the smell of rotting flesh, in particular human flesh. Maggots do, they love it, so they are attracted to it, and actually they are probably there even before this active decay. Many blowflies are known as the first witness to death because they're attracted almost instantly, and so they may have been there quite a while. Actually, before the maggots can pupate, they need to have been there a while. So the corpse here is reduced to about 20% of its original mass because, as we said before, the body has leaked fluids in that second stage known as bloating. So by active decay, the corpse is rapidly losing mass and going down to about 20% of its original mass. Okay, and so here's another warning. We're going to have a fairly graphic picture in a moment, so if you'd like to look away or close your eyes, I would advise you to do so now. Okay, so now quite a bit different from that bloated hand that we saw in the bloating stage. now we have active decay, and again, you can see the skin ruptures and just the appearance of, very decayed flesh that's undergoing rapid active decay. All right, and for at least a moment, we have no images on the screen, so you're welcome to look back if you had looked away or closed your eyes. The next of our stages of decomposition is advanced decay, and so at this stage, the body is really now just reduced to hair, skin, and bones, so very much a shell of the former human. We have a fly population that's been reduced now because there's relatively little for the flies to feed on, but what we will often find at this stage is other arthropods like beetles would come and feed on the remaining tissues, or predator arthropods that feed on flies or beetles that are already at the body would show up around this stage. So this is the point where the flies have done most of what they can do, and so now they're leaving or being eaten by other arthropods that are feeding on the remaining tissues and the other insects present. And here we go, another warning about a graphic picture coming up, so if you'd like to close your eyes or look away, I suggest you do so now. All right, and so here we have an image of advanced decay. This is not a human, but it is an animal in advanced decay, and you see there's very little left at this point, really just the fur, the bones, and some skin. So only the toughest parts of the animal are remaining at this particular point of advanced decay. All right, and so we're back to no images and on to our final stage of decomposition known as dry remains. So at this point, we are down to the skeleton. So depending on the environment, temperature, humidity, etc., it can take longer or shorter to get to this point, but ultimately this is the final stage of decomposition. Most anything that can be decomposed by the action of insects or bacteria has done so, and we're left with just the most durable remains of the individual. So this is our final of the 5 stages, known as dry remains. Okay, and a final warning here, we've got a graphic picture coming up, actually a couple graphic pictures. So if you'd like to close your eyes or look away, I'll let you know when you can look back. If you're fine with watching the images, then by all means, keep your eyes open and face forward. All right, and so here we see again, it's an animal, not a human, but we're at the stage of dry remains. Really, it's pretty much just the bones that we can see, and the bones have been picked clean by the decomposers, and so really at this stage we have just the dry bone remains in this image. Now, if you're still looking away, I would still look away myself if I could, because even though this is an illustration, it's still quite graphic, but it's nice because it shows the different stages. So working from right to left, we have the fresh stage that looks pretty much like normal human flesh, maybe a little more pallid from that pallor mortis starting, but fairly lifelike, right? Living type thumb there on the far right. As we move into the index finger, we've got now bloat, where the index finger is bloated because of those gases being produced by the anaerobic bacteria that are still alive under the skin. Moving into the middle finger, we see active decay. We have the maggots breaking through, and again, if we could smell this particular stage, that's when it's really particularly pungent, unpleasant odor. Moving on to the ring finger, we're now at a stage of advanced decay, so we don't see many maggots or insects at that stage, really just the insects that feed on the flies or the beetles or whatever's been there. And then finally, moving on to the pinky, we're at the dry remain stage, where pretty much only dry skin, cartilage, and bone would be remaining. So there's a single image that runs us through our full 5 stages of decay. Okay, so we're all set with images. If you've been looking away or having your eyes closed, please feel free to look back. We'll discuss the types of mortis, right, from the Latin mors, mortis, meaning death. We've already mentioned a couple of these, but we'll go ahead and discuss all 4 of the different mortises. Again, livor mortis and pallor mortis go hand in hand, but there's again the 4 different types: algor mortis, rigor mortis, and livor mortis / pallor mortis, which, as I've already said, go hand in hand. We've already had a little experience discussing. So let's take a look at these one by one and hopefully get a better understanding of what they are and when they occur after death for the human body. Okay, so the first one we'll discuss is algor mortis, and that's the loss of heat from a body. So humans are warm-blooded animals, so as we die, the warmth generated by our metabolism stops, so the body gradually cools. And this happens about an hour after death, and so the body will lose heat by about 2 degrees Celsius for the first hour, and then 1 degree Celsius per hour afterward until the body reaches environmental temperature. So if we're talking about a cold winter night in the Northeast, then the body will lose heat for quite a while. If we're talking about a warm summer day, then it won't take very long to reach that final environmental temperature. So the lower the body temperature, the closer the body temperature is to the environmental temperature, then the longer it's been since death and the less certainty there is. And I'll make another plug at the end of the lecture, but there's a separate video on what's called a nomogram or nomograph, depending on where in the world you are, and that's an application of algor mortis that will allow for that determination of time, post-mortem interval that's passed, and so that'll give you some practice with that concept when you watch that video. Okay, our next mortis that we'll discuss is probably the mortis you're most familiar with, rigor mortis, and that's the shortening of muscle tissue and the stiffening of body parts in the position at death or close to at death, right? Because it's actually a little while after death. It's as soon as 2 hours after death, but again, it doesn't become full and complete until around 12 hours after death, and then it starts to leave the body around 24 hours after death, so the body becomes flexible again. That's a misconception you'll see in a lot of the crime dramas on TV or in the movies where everybody's always in a complete state of rigor mortis, right? The body's stiff, and that's just something that they'll typically show. Again, if it's been less than 2 hours, the body won't even have started to stiffen up. If it's been less than 12 hours, you're not going to find the full body in rigor mortis, and after about 24 hours since death, the body releases the stiffening of rigor mortis and goes back to a more flexible state. So even though rigor mortis is one you've probably heard of, it's one you may have misconceptions about because of how it's typically presented in movies and television. Okay, after a little break, we are back to a graphic picture coming up, so those who would prefer to can look away or close their eyes, and I'll let you know when it's safe to watch again. Okay, so in this image we see an individual who must have been found between 2 and 24 hours after death, again assuming ambient temperature. That those numbers do vary if we're talking about very cold or very warm conditions, but at or around room temperature, we'd assume this is between 2 to 24 hours because there's at least still some rigor mortis in the body. And based on the appearance of the arm of this individual, it's most likely that he was sitting in an armchair, and that's what was supplying that support, and that's when the muscle contracted, the muscle tissue contracted and took on that appearance, the curvature of the armchair, and that set in as rigor mortis during the time frame in which the body was recovered. So if we were to go less than 2 hours, that arm would be flat on the table. More than 24 hours, that arm would be flat on the table, but because he was found between that 2 to 24 hour period, we see the arm in the position it was at the time the body was recovered. All right, so for at least a moment, you can open up or look back at the screen because we have no images here. Because they go together, we're going to discuss them together. So livor mortis and pallor mortis. Livor mortis, we've already discussed. It's where the blood settles to lower parts of the body due to gravity, right? The heart stops pumping, so we're no longer fighting against gravity, so gravity rules. Up until 8 hours, then it's permanent, and if the person's moved beyond that, then the livor mortis patterns don't change, and that could could be an indication that the body's been moved after the 8-hour mark. Pallor mortis, again, the body becomes pale in color because the blood that's been well circulated and giving that pinkish hue to the human body has now settled away according to gravity in what we call livor mortis, and that results in the body becoming pale in color, and that's known as pallor mortis. All right, so back to another little warning. We've got a graphic picture coming ahead, so look away or close your eyes right now if you don't want to see it. Okay, so here we see two different images. On the top, we see that the individual must have had something, maybe leg braces or just some object that was pressing up against the legs in those fleshy colored areas, and the remainder was pooling and giving that classic livor mortis pinkish purple color. And of course, the body must have been flipped over because we would not see that side that's lowest to gravity, so the body's been flipped over to reveal the livor mortis patterns. Likewise, we have in the lower image an individual who must have passed while laying on his arm, and so or some sort of swan or something, right? But it's much more logical to think that was his arm there. I'm just trying to inject some humor into a very serious and dark, disturbing topic, but he was lying on his stomach with his arm where we see that lighter paler color, and therefore the blood pooled around that, giving the pinkish purple appearance to the rest of his front side that he was lying down on. Okay, so again, going back to that idea of algor mortis, the time period between death and the discovery of the body is critical. And so rigor mortis, if we're in a complete state of rigor mortis, that gives us an idea. We're probably, around the 12-hour mark. If we've got livor mortis, we're between 2 and 8 hours. But what gives us the most accurate postmortem interval is algor mortis, is the loss of body temperature, and that allows us to determine an estimate of the time of death using the rectal temperature. The deep rectal temperature is the most reliable. It's the slowest loss. You don't want the skin temperature. That's far too rapid. So comparing that deep rectal temperature with the air temperature, taking into account the body weight and other factors. Is the body wet? Is the air moving or still? All these different factors. Is the body clothed? So please do go ahead and watch the nomograph video, and then be prepared. You're likely to see one question on exam 5 that asks about this topic of postmortem interval by nomogram. And I can't remember if I mentioned, but we didn't have any graphic photos, but we do have somewhat graphic photo here and in the next slide. But again, nothing that I think is going to be too upsetting for anybody. So if you've been looking away, please go ahead and look back or open your eyes, because I think these are pretty mild. So let's talk about a forensic autopsy. So we already said an autopsy is the medical examination of a body. It's performed by a forensic pathologist or a medical examiner who typically is a forensic pathologist, or some combination of the two, along with maybe an assistant. And not every person who dies has an autopsy, right? There has to be something suspicious or unnatural suspected about that death. And so this would be when foul play is expected. So there's two different parts. So you have the external examination, which involves photographing and examining the outside of the body. And then after that, which has been hopefully very well recorded, because once you start the internal exam, you're not going to be able to get back that original external appearance, because you're going to be doing lots of cutting and things. That will be the internal examination. Okay, so during that external examination, you're looking for features on the outside of the body that might give some indication as to how and why this person died, and if there's anything suspicious about that. So in particular, you're looking for lacerations, abrasions, bruises, puncture wounds. As I already mentioned, there's going to be a lot of photographing done, because once you start cutting, you're not going to be able to preserve this evidence anymore. If there's physical evidence on the body, so samples of hair, especially if it looks like hair that's not the victim's hair, that could be a suspect hair that could potentially link the responsible individual to this crime. Material under the fingernails, if the victim got in any defensive wounds by scratching the assailant. Other types of transfer evidence, right? Fibers or glass or anything that might be present, you're going to want to make sure that you collect. And then the body is cleaned and rechecked for wounds to see if anything else has become visible before the move to the internal examination. Okay, so the internal examination is just what it sounds like. You're going to inspect the internal organs for evidence of trauma or indications of death. Typically, you're going to be either looking at a T or a Y incision. So our picture shows a Y incision. A T incision would be just be a straight line along the top of the chest there. But in either event, the goal is to open up the body along the trunk there and to be able to remove the organs in sections corresponding to each of the cavities, weighing those organs, examining for any evidence of suspicious behavior. The scalp is pulled away from the skull, and an electric saw is used to expose the brain after, again, cutting from ear to ear to perform that brain examination. So in order to examine the brain, it has to be cut from the spinal cord and then lifted out for a full examination to see if there's any indication of any trauma or anything that may have been responsible for this individual's death from the examination of the brain. Now, because these are physicians that are performing these autopsies, they're going to be using anatomy terminology, right? So if we think about the human body, the axial portion is the head, neck, and the trunk, and then the appendicular portions would be the arms and the legs. So we see that in our image there where we've highlighted those two different parts of the body. So the main part, the axial part, head, neck, and trunk, and then the appendices, the arms and legs, being separate. All right, so I mentioned the examination involving a look at those cavities. So the body's divided into a number of different cavities. So we have the cranial cavity, and that one we've already discussed, where the brain resides, and the brain would be removed for examination from the cranial cavity. We have the dorsal cavity, which is where the bones of the skull all the way down through the spinal column. That's our dorsal cavity. There's the ventral cavity, which is on the anterior side, right? So the front side of the individual. It includes the thoracic and abdominopelvic cavities. Then we have, again, the thoracic cavity itself is the chest area, which is a subgroup there of that ventral cavity, which would include the esophagus, trachea, lungs, heart, and endocrine glands. We have the abdominopelvic, which is the other part of the ventral cavity, and that is the abdomen and pelvis. And within that, if we think about just the abdominal part of the abdominopelvic cavity, we've got the stomach, liver, gallbladder, pancreas, spleen, kidneys, intestines, and for females, ovaries. So lots of things there in the abdominal cavity. And then in the pelvic cavity, we have the colon, rectum, bladder, uterus for females, male genitalia for males. And finally, the diaphragm is that thing that separates the thoracic and abdominal pelvic regions. So that's how we can make that distinction. All right, so lots of different cavities and lots of different features within those cavities. Now, following along with that anatomical terminology, we have the transverse plane, which gives us that horizontal view, the top side and the bottom side of the body, sort of splitting us in the middle. We've got the sagittal or medial view, which splits the person down the center in a mirror plane, where your left and right portions would be mirror images of one another. And then finally the coronal, which would give a frontal and a rear view if you were to split the person along that plane. So I don't expect any of you to have taken college level anatomy and physiology. So we're just trying to make sure we all have a reasonable basic understanding of these terms and how anatomical terms relate as they're needed in forensic investigations. Okay, so just a few more anatomical terms. So superior means above, inferior means below. Those should be pretty logical. They're similar to their everyday usages, although again, specifically here we're talking about the human body relative positions. We've got anterior, which is the front side, and posterior is the back side. And then finally, medial would mean closer to that midline, that sort of imaginary middle of the body that we have there pointed out on our picture. And then lateral would be further away from that midline of the body. So there's a number of different terms and their definitions, and hopefully they make sense. If not, certainly please reach out if you would like more explanation or just need a little bit of additional explanation. Okay, now to round it out, we've got our final terms here for the anatomical terminology, and that would be proximal, which would be closer to the trunk of the body. Distal would be the opposite, further from the trunk. We have superficial, which means close to the surface, and the opposite of that would be deep, far from the surface. And then finally, cranial, close to the top of the skull, right, the cranium. And caudal, which would be towards the bottom of the body. So those are our lists of anatomical terms, and hopefully they all make sense. They should sound similar to a lot of everyday words that you know and understand, so hopefully they're not too much trouble. But if they are causing you any difficulty, please do reach out for some assistance. Okay, so now let's switch gears from forensic pathology to forensic anthropology. And so this is an area where I can sympathize with Sir Arthur Conan Doyle, the creator of Sherlock Holmes. Conan Doyle was a bit jealous of Holmes, his creation, because Holmes became more famous than Conan Doyle himself. Well, I'm guessing some of you out there are probably familiar with the show Bones and the Dr. Brennan from that show. And she's certainly far more famous than I am, even though she's fictional and I'm real. So again, I can have a little jealousy there. Can't watch the show, and it's not just the bad acting. It's just that I can't get over that Dr. Brennan similarity. So back to the meat of it. Forensic anthropology involves the examination of human skeletal remains or decomposed bodies. Usually they've been buried and they've been there for some time and quite decomposed, so well beyond the first 4 stages we talked about, so pretty much the dry remains stages of decomposition. In order to become a forensic anthropologist, again, similar to a forensic pathologist, you'll have the 4 years of undergraduate study, but instead of medical school, typically we're looking at another 3 or 4 years for a master's degree, and then 3 to 5 years for PhD. Again, it varies. You don't need the master's degree necessarily, so maybe you go right to the PhD and get that in about 5 years, or maybe you do the master's in 2 or 3 years and another 3 years or so for the PhD. It's all depending on if you have to work outside or if you're raising a family or any of the things that can interfere with anyone's education. What can a forensic anthropologist do? Well, they can help to determine the sex, the age. The postmortem interval here is going to be quite a bit longer than the postmortem interval that we'll discuss in that video on nomograms. That's only good for a few days after death. This would be generally a long time after death, weeks, months, years. So it's going to be quite a different postmortem interval, but still helpful. From the skeletal remains, race and cause of death can also typically be determined in many cases. Okay, so forensic anthropology itself really got established in 1878 by Thomas Dwight, and so therefore he's known as the father of anthropology. He published an essay on how to use the parts of the skeleton to establish things like age, sex, height, all the things that we just mentioned previously, and so he gets a lot of the credit, at least in Western history, for developing this field that's become forensic anthropology. Now, nearly 100 years after Thomas Dwight established the field, we have Dr. William Bass, who created a body farm at the University of Tennessee in 1971. And so what that body farm is, it's a closed off area that contains many corpses. I'm assuming these are individuals who have donated their bodies to science, and they've been laid across the land at different stages of decomposition so that people can learn what decomposition looks like after a given amount of time. So this is really an important step in better understanding decomposition, and a very worthwhile project that Dr. Bass has undertaken. So it's made him quite a famous forensic anthropologist, and it's helped tremendously with understanding the visual aspects and the scientific details of decomposition at various points in time. All right, so again, we've got a lot to learn from these skeletal remains. So what are some things that the remains can tell us? Well, first of all, are we looking at human skeletal remains or not? Are we looking at some other animal, even a primate that may have a very similar appearance to human skeletal remains, can be distinguished by those different features. The age of our person, if we've determined it is indeed a human person, an approximate age can be determined based on bone length and bone density and things like that. Sex, in particular, from bone density and from things like the width of the pelvis, if we happen to have that intact, or if we can put it back together, if we have all of the pieces. The height of the individual from the long bones. The race, we'll talk about the eye orbit and how that can help in determining race of our human. Injuries, has the person had any broken bones or bullets that have injured the bones, things like that. How many victims do we have? How many bones are present? Are we looking at some sort of mass grave, or is there just a single victim, or do we have just a couple of victims? How long have the bones been at the scene? Again, this would be something that the body farm that Dr. Bass has would help to identify. And again, also we have to take into consideration things like, are we in a seasonal area where it gets significantly colder in the winter and warmer in the summer, or are we someplace where it's fairly constant temperature year-round? All that goes into determining the length of time the bones have been at a given scene. Okay, so looking at determining age from those skeletal remains. So we've got the width of sutures, right? The sutures are these zigzag cracks on the skull that have fused together since infancy, right? You've got your soft spot when you're baby just born, right? That allows the parts of the skull to move in to make childbirth a little easier. Once we're out of the womb, then we don't need that anymore. It starts to fill in, usually takes close to 2 years or so for that to fuse, and then beyond that time, the fused lines, those zigzag cracks, become less and less visible as the person ages. And so if the a particular body under examination has notably visible zigzag cracks, then it's a younger person. If it's almost invisible zigzag cracks, then it's an elderly individual who's had a long time for those fused lines to become less and less visible. And I mentioned this a little bit, but we can typically determine gender from the skeletal remains. So we've got males, or at least sex. Males have long pelvic openings. They're typically narrow, and long and narrow sacrum. The skull is typically larger in males than females. The brow and jaw tend to have a square appearance. And it's a fairly robust skeleton, so it's fairly good bone density and larger in size, whereas females tend to have large circular pelvic openings because they're the ones that are going to be responsible for childbirth. So evolutionarily, they've evolved to have the larger, more circular pelvic opening because they're the ones that can carry children for the female sex compared to the male sex. You've got a wide sacrum, a smaller skull, a rounded brow and jaw as opposed to the squarish brow and jaw for the male. And finally, it's a more slender skeleton overall compared with the more robust skeleton of the male. So that's typically how the sex can be determined from skeletal remains. Okay, now let's consider how we might be able to determine race from skeletal remains. So those of Asian and Native American descent tend to have flat or projected outward frontal planes on their skulls. There's a small rounded nasal cavity and circular eye orbits. So those tend to be the key characteristics to associate a person's remains with Asian or Native American descent. If the individual is of European descent, then we're expecting to see a flat cranium, a long, narrow nasal cavity, aviator-style glasses, which would be sort of oval eye orbits that are a little bit rectangular at the top. And then finally, for those of African descent, we'd expect the cranium projected outward, wider nasal cavity compared with the other two races we discussed, and eye orbits that are more squarish or rectangular in appearance compared with the Asian or European descent individuals. All right, so far talking about things like sex and race, those are class characteristics. How about identifying an individual from skeletal remains, getting down to not only just the race or the sex, but the specific person that we might be looking at? Well, forensic anthropologists can use software to make facial reconstructions that can give surprisingly detailed representations of how that person looked just from the skeletal remains. Identifying past surgeries or broken bones that might help identify a person individually based on their medical history. And finally, dental records. The teeth are even more robust than the bones, and so perhaps by comparing those teeth found on the individual at the scene, comparing those against dental records could be a valuable way to identify the individual. Now, it would require them having dental records on file, so just be careful. It's not always possible from the teeth to be able to determine identity from dental records, but oftentimes it is possible. Now, following along on that line of thought, if we do have the teeth present, then the individual that we would need to consult is no longer a forensic anthropologist, but now is the time for a forensic odontologist. So this is a forensic dentist, so someone who's done 4 years of undergraduate work, 4 years of dental school, and then a specialized residency of 1 year or 3 in forensics. So they can use those dental records to identify victims based on the teeth that are present in the skeletal remains. They can estimate the age of the individual based on, again, if the person has, wisdom teeth or still juvenile teeth or things like that. They can even examine the teeth for DNA to get individualized evidence. If we've got bodies that are severely burned, disfigured, decomposed, right? The teeth are more robust than the bones, so they may have survived better and they may give a better likelihood of being able to identify that individual than the bone record that the forensic anthropologist might be able to acquire. And finally, we can identify suspects by matching bite marks. This is problematic. This is where science has just finally caught up with the hype, and there were even some FBI experts who may have overstated what could be done by bite mark analysis. Really, bite marks are better to exclude suspects than to necessarily identify them individually. If there's an unusual bite pattern, then that certainly could give rise to individual identification, but there's a lot of similarities in bite patterns, so you have to be very careful before you use bite marks to unequivocally identify an individual. But in the case of Ted Bundy, a famous American serial killer, it was bite marks that were one of the key pieces of physical evidence that ultimately got him convicted. And again, even in that case, it may have overstepped the bounds, but certainly he was someone who had gotten himself out of situations in the past. He was a law student. He argued his own defense at times, and so the bite marks being so critical in that case were probably enough to sway the jury for the conviction that finally put him behind bars. We discussed earlier the idea of the blowfly being attracted to dead tissue, and then the beetles and other insects also coming, the beetles for the skin, and then other predator insects that eat the flies and the beetles that are attracted to the decaying flesh. The forensic entomologist would be the professional who would be the expert in this area, because an entomologist studies insects, and so a forensic entomologist would have specialized knowledge with the forensic application of this. And again, we're going to be similar to an anthropologist, probably 4 years of undergraduate, 2 or 3 or 4 years to a master's, maybe another 3 to 5 years to a PhD, and the forensic entomologist would be able to discuss the life cycle of the blowfly, especially, present as an expert witness in a jury trial. So what the forensic entomologist would probably testify to is that the blowflies will lay their eggs in open wounds or orifices, right, places that are a little bit protected, and allow for access to nutrients from the decaying body. So the eggs then hatch and produce larvae known as maggots. The maggots will shed their skin 3 times. It's called an instar, 3 instars, and then finally form a hard shell called a puparium. And that's when the maggot undergoes metamorphosis to have a very different appearance and emerge as an adult blowfly, right? Sort of the same process as what a butterfly undergoes as it goes from being caterpillar to butterfly, but again, not so celebrated. We tend not to care as much about the metamorphosis of a maggot to fly as we do caterpillar to butterfly. So that adult blowfly can either fly away and try to find new food, or stick around and make a second or third or fourth or fifth generation of flies. As we mentioned, eventually the fly population will go down because the body will decay, progress through those different stages of decay so that there's not too many nutrients left. And temperature and weather certainly affect this. If we're talking about the Northeast in the cold winter, that life cycle is going to be slowed. And if we're talking about tropical areas, that life cycle is going to speed right up. So different things to keep in mind there as we talk about forensic entomology. And so here we see a picture of that blowfly life cycle. So the mommy fly and the daddy fly find the body and get married, and then lay the eggs. And about 23 hours after those eggs have been laid, emerges that first instar, the first maggot. The maggot, after about a day, 27 hours or so, it sheds its skin and becomes the second instar. And then in another 22 hours, sheds again to become the third instar, the largest maggot. And that's the maggot that is going to eat up, and then after about 130 hours, go into the puparium for another 143-ish hours before emerging as an adult blowfly and starting the whole process again. So the times here are temperature dependent, weather dependent, right? If it's a dry area, if it's a humid area, those can have impacts. So they're ballpark figures, but they do tend to be pretty well representative of the blowfly life cycle. Okay, now finally, before we wrap things up here, let's consider how we would undergo an excavation of skeletal remains if this is a buried individual. First of all, we'd want to remove any debris. Lime in particular is likely if the person has been buried in the earth. Vegetation may be present, right, if there's plants or decaying plant matter around the body. So tape off and sketch the excavation area. We want detailed records. We don't want any more disturbance. Unfortunately, if it's a fairly shallow grave, animals may have already disturbed the individual, the skeletal remains of that individual to some extent. But if it's a deeper grave, then maybe the skeleton will be in fairly good shape. Next, if we're removing soil, make sure to sift each layer to collect any evidence that might be present along with the soil. We don't want to lose that. Next, document with photos, sketches, maps, inventory, recording those measurements. Good record keeping is oftentimes the difference between conviction and the failure to convict an individual. So you want to make sure that you have as solid a case against the suspect as possible, because again, in cases of skeletal remains, we can't help that victim, but we can certainly help get justice and get the family the closure they might need, and things like that. Finally, package each bone individually and submit it for analysis at the crime lab, or again, if we've got a situation like this, we may have the forensic anthropologist actually in the field to help with that particular stage. So that's how we would go about excavating human skeletal remains. All right, so to wrap it all up, we have a number of different forensic professionals that investigate human remains. We talked about the forensic pathologist, the forensic anthropologist, the forensic odontologist, forensic entomologist. So lots of different people who study these remains and try to use their expertise to identify victims and get physical evidence to link suspects and bring closure to families, and all wonderful things, and very skilled and dedicated people who do that work. Forensic pathologists and forensic anthropologists are different, right? The forensic pathologists are MDs with specialized training. The anthropologists tend to be PhDs with, again, their expertise on the skeletal end of things. So although forensic pathologists tend to be involved in more recent deaths and anthropologists tend to be involved in longer postmortem intervals, they both focus on death and decomposition, and they can both help define that postmortem interval. For the forensic pathologist, it's going to be typically pretty short, hours, days. For the forensic anthropologist, it's going to be quite longer, probably months, years. But they both have those common bonds among them. Finally, don't forget, please go ahead and view that other lecture video. It's in the Blackboard Unit 12 folder, or you can link to it right here on using a nomogram or nomograph. Again, both terms are used to find postmortem interval. There's also some additional practice problems and the answer keys in the Unit 12 folder. And last but not least, thank you very much for a great semester. I hope you've enjoyed all of these topics, and I hope that you've enjoyed the course overall, and certainly would welcome any feedback you might have. So please don't hesitate to reach out to me. Thank you for a great semester, and good luck in all your future endeavors.